Hello, my dear future doctors. Welcome to Kerala NEET Academy. It's me, Dilip, your biology faculty. Today we are going to learn the chapter Microbes in Human Welfare. What microbes are exactly? Microbes, they are called microorganisms. They are the tiny living organism that can be too small to be seen with the naked eyes. Example for microbes are bacteria, fungi, virus, protozoans. These microbes exist in everywhere. They are found in soil, water, then air, some extreme climatic conditions within human body. And these microscopic forms play a crucial role in nature, human body, and various industries. Okay, the microbes can be harmful or beneficial. In this chapter, we are going to discuss the beneficial role of microbes. That is, microbes in human welfare. Clear? These are the subtitles we are going to discuss. Microbes in household product, microbes in industrial products, microbes in sewage treatment, microbes in biogas productions, and microbes used as biocontrol agents, and microbes in biofertilizers. So first we are going to discuss microbes in household product. These are some important microbes that are used in the production of some household product. The first one is lactobacillus or lactic acid bacteria. Simply we can say lab. Okay, so lactobacillus or lactic acid bacteria, they are used to convert milk into curd. Okay, so lactobacillus or lactic acid bacteria are used to convert milk into curd. Some bacteria are also used for fermenting dough to make foods such as dosa, idli, etc. Okay, some bacteria are used for fermenting dough to make foods such as dosa, idli, etc. And Saccharomyces cerevisiae is a fungus. It's a yeast. It's a common yeast. Saccharomyces cerevisiae. Saccharomyces cerevisiae is also called baker's yeast, used to make bread by fermenting dough. It's very important. Saccharomyces cerevisiae. It is called baker's yeast, used to make bread by fermenting dough. Some microbes are also used to ferment fish, soybean, bamboo shoots, and to produce foods. Okay, you know the toddy, the traditional drink used in southern part of India, that is toddy. Toddy is made by fermenting sap from palms. Toddy is made by fermenting sap from palms. Okay, so cheese, it's a very old food items in the world, cheese. See, there is a bacteria called Propionibacterium shermani. It's a very, very important bacteria called Propionibacterium shermani is used to make Swiss cheese. Okay, and there is a fungus called Penicillium roqueforti. Penicillium roqueforti. It is used to produce Roquefort cheese. Okay, so very, very important. You can match the following chance. So lactobacillus or lactic acid bacteria convert milk into curd. Okay, then Saccharomyces cerevisiae. It's a fungus. It's a yeast. It's also called baker's yeast, fermenting dough to make bread. Okay, and Propionibacterium shermani. Study the name. Propionibacterium shermani is used to make Swiss cheese. And there's a fungus called Penicillium roqueforti. Penicillium roqueforti is used to produce Roquefort cheese. Okay, so this is a Swiss cheese. You can see the large holes. This large holes is due to the production of carbon dioxide. This is a Swiss cheese, and this is a Roquefort cheese. Here we are using Propionibacterium shermani. Here we are using Penicillium roqueforti, a fungus. So next we are going to discuss microbes in industrial product. Microbes in industrial product. Say production of beverages, antibiotics, production of beverages, antibiotics, etc. on an industrial scale requires growing microbes in a large vessels. These vessels are called fermenters. we can say these are the large vessels fermenters. Okay, so production of beverages, antibiotics, organic acids in large scale, we need some large vessels. These large vessels are called the fermenters. Okay, they are called fermenters. First one, production of fermented beverages. For the production of fermented beverages, we are using Saccharomyces cerevisiae. It is also called Brevos yeast, Baker's yeast, Brevos yeast. Rendume Saccharomyces cerevisiae. So this Saccharomyces cerevisiae is used for the production of fermented beverages. Fermented beverages. Okay. There are two types of fermented beverages. Distilled and non-distilled. Distilled and non-distilled. Non-distilled. Example for non-distilled are wine, beer. So these are example for non-distilled. Whiskey, brandy, rum, gin, arak, all these are example for distilled fermented beverages. Okay. So Saccharomyces cerevisiae, it is also called Brevos yeast, are used for the production of fermented beverages. There are two types of fermented beverages, distilled and non-distilled. Wine, beer are example for non-distilled beverages. And whiskey, brandy, scotch, rum, gin, these are example for distilled beverages. So Saccharomyces cerevisiae. Next, antibiotics. What do you mean by antibiotics? Antibiotics are chemical substances produced by some microbes and can kill or retard the growth of pathogens. So they are against the life of pathogens. So they are called antibiotics. For humans, they are called pro-life. So antibiotics, they are chemical substance produced by some microbes and can kill or retard the growth of pathogens. Okay. So antibiotics are chemical substances produced by some microbes and can kill or retard the growth of pathogens. So antibiotics are also called antibacterials. Antibiotics are also called antibacterials. Generally, these are the drugs used to kill bacteria. Okay. Generally, these drugs are used to kill bacteria. So these antibiotics are also called antibacterials. Okay. See, the penicillin is the first antibiotic discovered by Alexander Fleming in 1928. Okay. So the first antibiotic is penicillin. The first antibiotic is penicillin. It was discovered by Alexander Fleming in 1928. It is obtained from a mold. It is obtained from a mold. Its name is called Penicillium notatum. Penicillium notatum. Penicillium notatum. Penicillium notatum. Clear? So first antibiotic is penicillin. It was discovered by Alexander Fleming in 1928 and it is obtained from a mold of fungus. It is called Penicillium notatum. Ernst Chain, Howard Florey established its full potential as an effective antibiotic. Fleming, Chain, and Florey were awarded Nobel Prize in 1945. 1945. It's very important. So first antibiotic, penicillin, discovered by Alexander Fleming. It is obtained from the mold Penicillium notatum. Its full potential as an antibiotic was established by Ernst Chain, Howard Florey, and Fleming, Chain, Florey were awarded Nobel Prize in 1945. Okay. And we also used antibiotic to treat plague, whooping cough, diphtheria, leprosy. We are using antibiotic to treat plague, whooping cough, diphtheria, leprosy. Some other examples for antibiotics are azithromycin, then ampicillin, tetracycline. Okay, all these are examples for antibiotics. Next, we are using for, we are going to explain the production of organic acids. Okay, so we are using some microbes to produce some acids. See, Aspergillus niger, it's a fungus. Aspergillus niger is a fungus used to produce citric acid. Acetobacter aceti is used to produce acetic acid. It's a bacterium. Clostridium butylicum. It's a bacterium used to produce butyric acid. Okay. Lactobacillus is a bacterium used to produce lactic acid. It's a very important table. So microbes are used to produce some organic acids. Okay. So the table is very important. Match the following chance. Aspergillus niger. It's a fungus used to produce citric acid. Acetobacter aceti. Acetobacter aceti is a bacterium used to produce acetic acid. Clostridium butylicum. It's a bacteria used to produce butyric acid. Lactobacillus, it's a bacterium used to produce lactic acid. Okay. So for the production of fermented beverages, we are using Saccharomyces cerevisiae. Okay. So first antibiotic is penicillin. It is obtained from the mold Penicillium notatum. It was discovered by Alexander Fleming. Okay. Chain, Flore and Fleming awarded Nobel Prize in 1945. Okay. So next, microbes are used for the production of some organic acids. Aspergillus niger, a fungus used to produce citric acid. Acetobacter aceti, a bacterium used to produce acetic acid. Clostridium butylicum, a bacteria used to produce butyric acid. Lactobacillus, a bacterium used to produce lactic acid. For the production of alcohol, we are using the yeast, which is Saccharomyces cerevisiae. We already know is to produce ethanol. Okay. So Saccharomyces cerevisiae is also used to produce ethanol. And enzymes, very, very important enzymes. Okay. Lipase. Some microbes produces lipase, enzyme lipase. And these lipases are used in detergent formulations. It helps to remove oily stain from the laundry. Oily stain from the laundry. It's very important. Some microbes, some fungus produce lipases. The name is not mentioned in your NCERT. Okay. Some fungus produces lipases enzyme. These enzymes are used in detergent formulation, which is helped to remove oily stain from the laundry. Dress-ilakku edavadu oil stain ange irunnalu ange dirt irukalla chance adhigama irukkum. So and the oil ne remove pannan we are using the enzyme lipase. And this lipase is produced by some fungus. Like that, we are using the enzyme pectinases and proteases. They are used to clarify bottled juices. Okay. So lipases are used in detergent formulation, helps to remove oily stain from laundry. And pectinases and proteases, they are used to clarify bottled juices. നമുക്ക് தெரிയും அந்த ഷോപ്പിൽ നിന്ന് കിടക്കുന്ന அந்த ബോട്ടിൽഡ് ജ്യൂസിലൊക്കെ கொஞ்சம் ജ്യൂസ് എല്ലാം നല്ല ക്ലിയറാக തന്നെ இருக்கும் അത്. ഓക്കെ. റീസൺ அங்கே எந்த என்சைமினை നമ്മൾ யூஸ் பண்ணணும்? pectinase and proteases. Okay. So these enzymes are used to clarify bottled juice. Next enzyme is called streptokinase. It's a very, very important streptokinase. Streptokinase is produced by the bacteria Streptococcus. Okay. So streptokinase is an important enzyme produced by the bacteria Streptococcus. And this streptokinase is used as a clot buster. It's used as a clot buster to remove a clot from the blood vessels of patients who have a myocardial infarction. It's a very, very important. Okay. So which enzyme is used as a clot buster? Streptokinase. It is taken from the bacteria Streptococcus. And this streptokinase used as a clot buster to remove clots from the blood vessels of patients who have myocardial infarctions. Okay. Especially നമുക്ക് தெரியும் அந்த coronary artery ഒക്കെ இருக்கு. அந்த coronary artery-ல ஏதாவது block ഒക്കെ വരുന്ന ടൈമിൽ we can use this streptokinase enzyme. This enzyme remove the clot from the blood vessels. Okay. So this is the enzymes. So pectinase and proteases, they are used to clarify the bottled juice. Okay. Lipases are used in detergent formulation to remove oily stain from the laundry. Clear? Yes. Next, statin, some bioactive molecule. Yeah, next one more there, cyclosporine. It's a very, very important cyclosporine. It is produced by a fungus Trichoderma polysporum. It's a very, very important. Cyclosporin A is produced by a fungus Trichoderma polysporum. It's a very important part. So cyclosporin A is produced by the fungus Trichoderma polysporum. It is used as an immunosuppressive agent in organ transplant patients. നമ്മുടെ ഓർഗനൊക്കെ ട്രാൻസ്പ്ലാന്റ് பண்ணுന്ന டைமில் அந்த ரெசிபியன்ட் இம்யூன் சிஸ்டம் அந்த ஆர்கனை ரிஜெக்ட் பண்ணாനുള്ള சான்ஸ் அதிகமாக இருக்கும். ஓகே, யூனோ த கிராஃப்ட் ரிஜெக்ஷன். ஓகே, ஸோ அந்த கிராஃப்ட் ரிஜெக்ஷனை ப்ரிவென்ட் பண்ணணுங்கில் we have to suppress the immune system of the recipient. For that, we are injecting which substance? Cyclosporin A. So cyclosporin A, it is produced by Trichoderma polysporum, used as an immunosuppressive agent in organ transplant patients. Clear? Yes. Next, statin. Statins are produced by yeast Monaskis purpurus. Very, very important. Okay, statin is produced by Monaskis purpurus. It's an yeast. And statin is used as a cholesterol-lowering agent. It inhibits the enzymes responsible for the synthesis of cholesterol. Okay, very, very important. So statin is produced by Monaskis purpurus. It's an yeast. So this statin is used as a blood cholesterol lowering agent. Blood cholesterol lowering agent. It inhibits the enzyme responsible for the synthesis of cholesterol. It acts as a competitive inhibitor. Statin acts as a competitive inhibitor of the enzymes responsible for the synthesis of cholesterol. Okay, so microbes are used for producing organic acids, then ethanol, then some enzymes and some bioactive molecules. Okay, so Aspergillus niger is a fungus used to produce citric acid. Acetobacter aceti is a bacterium used to produce acetic acid. Clostridium botulicum is a bacteria used to produce butyric acid. Then Lactobacillus is a bacteria used to produce lactic acid. And Saccharomyces cerevisiae is used to produce ethanol. And some microbes are used to produce lipases. And these lipases are used in detergent formulation to remove oily stain from the laundry. Next, the two enzymes, proteases and pectinases, they are used to clarify bottled juice. And there is another important enzyme called streptokinase. It is taken from the bacteria Streptococcus. And this streptokinase is used as a clot buster to remove blood clot from the blood vessels. They remove the clot from the blood vessels. And next is cyclosporin A. It's a very important. Cyclosporin is an immunosuppressive agent. It is produced by a fungus called Trichoderma polysporum. Trichoderma polysporum. And there is another compound called statin. Statin acts as a cholesterol lowering agent. Statin acts as a cholesterol lowering agent. And this statin acts as a competitive inhibitor of the enzyme responsible for the synthesis of cholesterol. Okay, this part is very, very important for your NEET exam. Match the following chance. So microbes in sewage treatment. What do you mean by sewage? Municipal wastewater is called sewage. Okay, so municipal wastewater is called sewage. Municipal wastewater which contain large amount of organic matter and microbes. Municipal wastewater is called sewage which contain large amount of organic matter and microbes. Sewage is treated in sewage treatment plants or STPs. This sewage is treated in sewage treatment plants to make it less polluting. It includes two stages. First one is called primary treatment and second one is called secondary treatment. So what is sewage? Municipal wastewater is called sewage, which contain organic matter and microbes. Okay, this sewage is treated in sewage treatment plants. Two important stages. First one is called primary treatment and second one is called secondary treatment. Primary treatment and secondary treatment. In primary treatment, it is a physical treatment. Inga nammal microbes ne onnume use pannala, so it is a physical treatment. It's a physical removal of particles. It includes sequential filtration and sedimentation. Very, very important. Primary treatment include, okay, two steps. First one is called sequential filtration. Here we are using net with a different mesh size. Okay, here we are using net with a different mesh size. So, on the madri sequential filtration, we are removing the floating debris. Second one is sedimentation. After sequential filtration, some soil and small pebbles are dissolved in the water. So, to remove this, next step is called sedimentation. Okay, so this is the sequential filtration. And second one is called as sedimentation. This is a primary treatment. A primary treatment le two step irukku. First one is sequential filtration. We are removing the floating debris. And second one is sedimentation. Here we are removing the grit. What is grit? Soil and small pebbles which are dissolved in the water. Okay. After the primary treatment, we get the primary effluent and the primary sediment. Okay, so the water is called primary effluent. And the sediment is called primary sludge. Okay, the settled solid form the primary sludge and the supernatant form the primary effluent. Supernatant means water. It's called primary effluent. Okay, this is called primary sludge. And this water, this is called primary effluent. We need the primary effluent. So this is a primary treatment. Secondary treatment, the primary effluent is passed into large aeration tanks and constantly agitated. Okay, and the primary treatment primary effluent and the water. That primary effluent is passed into large aeration tank and constantly agitating. What is the purpose of agitation? To increase the level of oxygen. Okay, so the primary effluent is passed into large aeration tank and constantly agitating. Okay, as a result, level of oxygen increases. This allows the vigorous growth of useful aerobic microbes into flocs. Okay, after primary treatment, the primary effluent is passed into large aeration tank and constantly agitating. So the level of oxygen increases, which allows the growth of useful microbes in flocs. What do you mean by flocs? Large bacterial mass associated with the fungal filaments. Okay, bacteria associated with the fungal filaments are called flocs. This can be framed as a question. What do you mean by flocs? Aerobic bacteria associated with the fungal filaments. Aerobic bacteria associated with the fungal filaments are called flocs. And these flocs consume the organic matter of the effluent. And the water organic matter in the flocs eat pannumadu. Okay, they are eating the organic, they are consuming the organic matter in the effluent. This reduces the BOD of the effluent. Very, very important. What is BOD? BOD is called a biochemical oxygen demand or biological oxygen demand. BOD is called a biochemical oxygen demand or biochemical oxygen demand. Okay. So after primary treatment, the primary effluent is passed into large aeration tank and constantly agitated. Due to that agitation, level of oxygen increases. Okay, so this allows the growth of useful aerobic microorganisms into flocs. What is flocs? bacteria with the fungal filaments. And these flocs consume organic matter in the water, thereby reducing the BOD. What is BOD? Biochemical oxygen demand or biological oxygen demand. So what is BOD? BOD is it is an amount of oxygen consumed by bacteria to oxidize all the organic matter in 1 liter of water. It's very important. See, BOD, it's the amount of oxygen. It's the amount of oxygen consumed by bacteria. It's the amount of oxygen consumed by the bacteria to oxidize, to consume, to oxidize all the organic matter in 1 liter of water. This is called BOD. Okay. It is a measure of organic matter present in the water. അപ്പം വാട്ടറിൽ ഓർഗാനിക് മാറ്റർ അധികമായിരുന്നാൽ ബിയോഡിയും അധികമായിരിക്കും അത്. ഓക്കെ, സോ ആൻഡ് ബാക്ടീരിയക്ക് നീഡ് മോർ ഓക്സിജൻ. സോ ഗ്രേറ്റർ ദ ബിയോഡി മോർ ഈസ് ഇറ്റ്സ് പൊല്യൂട്ടിങ് പൊട്ടൻഷ്യൽ. അപ്പൊ നമ്മൾ വാട്ടറിന്റെ ഒക്കെ ബിയോഡി ചെക്ക് പണ്ണുന്ന ടൈമിൽ அந்த ബിയോഡി വാല്യൂ അധികമാ இருக்கும். ഇറ്റ് ഇൻഡിക്കേറ്റ്സ് വാട്ടർ ഈസ് മോർ പൊല്യൂട്ടിങ്. ஓகே, ஸோ நம்ம பிஓடி செக் பண்ண டைமில் ஒரு வாட்டர்ன்ற பிஓடி கொஞ்சம் கம்மியாக இருக்குது. ஓகே, பிஓடி லோவாக இருக்குது, லெஸ்ஸாக இருக்குது. ஸோ நம்ம கன்ஃபார்ம் பண்ணால் அந்த வாட்டர்ன்ற பொல்யூஷனும் கம்மியாக இருக்கும் அது. ஓகே, ஸோ கிரேட்டர் த பிஓடி மோர் இஸ் இட்ஸ் பொட்டன்ஷியல், பொல்யூட்டிங் பொட்டன்ஷியல். இப்போ பிஓடி அதிகமாயால் வாட்டர் பொல்யூஷன் அதிகமாக இருக்கும், வாட்டர் பொல்யூஷன் அதிகமாக இருக்கும். பிஓடி கம்மியாயால் வாட்டர் பொல்யூஷன் லெஸ்ஸாக இருக்கும். ஓகே, பிஓடி அதிகமாயால் വാട്ടർ പൊല്യൂഷൻ അധികമായിരിക്കും, BOD കമ്മിയായാൽ വാട്ടർ പൊല്യൂഷൻ ലെസ്സാ இருக்கும். So BOD, it's a measure of organic matter present in the water. So this part is very important. So what is BOD? It's the amount of oxygen consumed by bacteria to oxidize the organic matter present in 1 liter of water. BOD indicate the measure of organic matter present in the water. So BOD അധികമായാൽ വാട്ടറിന്റെ പൊല്യൂഷൻ അധികമായിരിക്കും, BOD കമ്മിയായാൽ വാട്ടറിന്റെ പൊല്യൂഷൻ ലെസ് ആയിരിക്കും. ക്ലിയർ? യെസ്. സോ വൺസ് BOD reduce ആச்சு. ഓക്കെ, ആഫ്റ്റർ ദാറ്റ് the effluent is then passed into settling tank. So once BOD reduce பண்ணியால നമ്മൾ அந்த வாட்டர்னை எங்கேக்கு மாറ്റുம்? செட்டிலிங் டாங்க் இருக்கு, லார்ஜ் செட்டிலிங் டாங்க் இருக்கு. அந்த லார்ஜ் செட்டிலிங் டாங்கிലേക്ക് നമ്മൾ പാസ് பண்ணும்போது where the bacterial flocks are sediment. നമ്മൾ മുன்னாடி പറഞ്ഞ அந்த bacterial flocks எல்லாமே இங்க sediment ஆகும். ஓகே, லார்ஜ் செட்டிலிங் டாங்கிലേക്ക് നമ്മൾ ചേഞ്ച് பண்ணும். இங்க ஆரிட் செடிமெண்ட் பண்ணும்போது flocks. What is flocks? Microbes associated with the fungal elements, fungal filaments. மேலே யார் இருக்கு? வாட்டர் இருக்கு. ஓகே, எஸ். So this is settling tank. So the effluent is passed into settling tank where the bacterial flocks are sediment. Okay, and this sediment is called activated sludge. The sediment is called activated sludge. Okay, this sediment is called activated sludge. Activated sludge. A small part of activated sludge is pumped back into the aeration tank to serve as an inoculum. Okay, ഇതിൽ கொஞ்சം പാർട്ട് തിരുമ്പി നമ്മൾ എങ്കേക്ക് കൊടുക്കും? അത് നമ്മൾ அந்த സെക്കൻഡറി ട്രീറ്റ്മെന്റിൽ ഫസ്റ്റ് യൂസ് பண்ணி ஏரேஷன் டாங்க் இருக்கு. ஓகே, இதில் அந்த ஃப்ளாக்ஸிന്റെ கொஞ்சம் பாட்டு നമ്മൾ எங்கே யூஸ் பண்ணும்? இந்த ஏரேஷன் டாங்கில் നമ്മൾ യൂസ് பண்ணும். எப்படி யூஸ் பண்ணும்? ഇനോക്കുലമായിട്ട്. எப்படி യൂസ് பண்ணும்? ഇനോക്കുലമായിട്ട് യൂസ് பண்ணும் അത്. ഓക്കെ, സോ ദ പ്രൈമറി എഫ്ലുവന്റ് എങ്കേക്ക് മാറ്റുന്നു? ഏറേഷൻ ടാങ്കിലേക്ക് മാറ്റുന്നു, കോൺസ്റ്റന്റ്ലി അജിറ്റേറ്റ് പണ്ണുന്നു. എയറോബിക് മൈക്രോബ്സ് ഇൻ ഫ്ലോക്സ് കൺസ്യൂം ദ ഓർഗാനിക് മാറ്റർ, BOD reduces. വൺസ് BOD റെഡ്യൂസ് பண்ணிയാൽ നമ്മൾ அந்த വാട്ടറിനെ എങ്കേക്ക് മാറ്റാൻ? നാച്ചുറൽ ബോഡീസിലേക്ക്. அந்த എഫ്ലുവന്റിനെ എങ്കേക്ക് മാറ്റാൻ? നാച്ചുറൽ വാട്ടർ ബോഡീസിലേക്ക്. വി റിലീസ് നാച്ചുറൽ വാട്ടർ ബോഡീസ് ആൻഡ് വാട്ടർ ആഫ്റ്റർ ദാറ്റ് എങ്കിൽ அந்த ഫ്ലോക്സിനെ സെഡിമെന്റ് പടുന്നു, ആക്ടിവേറ്റഡ് സ്ലഡ്ജ് കിടക്കുന്നു. ആക്ടിവേറ്റഡ് സ്ലഡ്ജിനെ നമ്മൾ എങ്കേക്ക് മാറുന്നു? അനൈറോബിക് സ്ലഡ്ജ് ഡൈജസ്റ്റേഴ്സിലേക്ക് മാറ്റുന്നു. ഓക്കെ, இந்த അനൈറോബിക് സ്ലഡ്ജ് ഡൈജസ്റ്റേഴ്സിൽ ആര് இருக்கு? கொஞ்சம் അനൈറോബ്സ് இருக்கு, அனேரோப்ஸ் ஆயிடுள்ள மைக்ரோப்ஸ் இருக்கு. அந்த அனேரோபிக் மைக்ரோப்ஸ் இந்த ஃப்ளோக்ஸ்னு எல்லாத்துக்குமே கன்சியூம் பண்ணுவது. So the anaerobic bacteria present in the anaerobic sludge digester, they digest the sludge and thereby we can produce the biogas. So this is a secondary treatment overall steps. Okay. So we will revise the topic. What is sewage? Municipal wastewater is called sewage, which contain microbes and organic matter. Okay. Sewage is treated in sewage treatment plants. இங்க ரெண்டு ட்ரீட்மெண்ட் இருக்கு, பிரைமரி ட்ரீட்மெண்ட்டும் இருக்கு, செகண்டரி ட்ரீட்மெண்ட்டும் இருக்கு. பிரைமரி ட்ரீட்மெண்ட்ல sequential filtration work, and then remove the floating debris. Second one is called sedimentation. We are removing the grit. Okay. We got two fractions. The sediment is called primary sludge and the supernatant is called primary effluent. Then the primary effluent is passed into large aeration tank. Large aeration tank where we are constantly agitating. As a result, level of oxygen increases, which support the growth of aerobic microbes, aerobic bacterium with fungal filaments called flocs. These flocs consume organic matter in the water. As a result, BOD reducing. Okay. Once BOD is reduced, we are passing this effluent into large settling tank where flocs are sedimented. These sediments are called activated sludge. And mele kadakkinna effluent we are released into water bodies. Okay. Then the activated sludge is passed into anaerobic sludge digesters. അതിൽ కొంచెം പാട്ട് തിരുമ്പി എங்கே കൊടുക്കും, എയറേഷൻ ടാങ്കിലേക്ക് കൊടുക്കും as inoculum. So the anaerobic sludge digesters contain some anaerobic microbes. They digest the flocs. From there we will get the biogas. Clear? This is a sewage treatment. It's very important. The Ministry of Environment and Forest has initiated Ganga Action Plan and the Yamuna Action Plan to save from water pollution. Okay, to save this river from water pollution, the Ministry of Environment and Forest, important. The Ministry of Environment and Forest has initiated Ganga Action Plan and the Yamuna Action Plan to save this river from water pollution. Clear? Yes. So next, microbes in the production of biogas. Microbes in the production of biogas. What is biogas? See, biogas is a mixture of gases, mainly methane, produced by microbes such as methanogens. Important. So biogas is a mixture of gases, mainly methane, produced by microbes such as methanogens. Methanogens grow anaerobically on cellulose material and produce methane along with the carbon dioxide and hydrogen. An example for methanogens are methanobacterium. Okay. So example for methanogens are methanobacterium. And the methanobacterium is found in anaerobic sludge and rumen of cattle. But രണ്ട് പ്ലേസിൽ വി ക്യാൻ സീ ദ മെഥനോബാക്ടീരിയം. ഒന്ന് നമുക്ക് എங்கே பார்க்க முடியும், നമുക്ക് അനേറോബിക് സ്ലഡ്ജ് ഡൈജസ്റ്റേഴ്സ് നമുക്ക് தெரியும் அது. அந்த செகண்டரி ட்ரீட்மெண்ட்டில் நம்ம அந்த ஆக்டிவேட்டட் ஸ்லட்ஜினை டைஜஸ்ட் பண்ணிய அனேரோபிக் ஸ்லட்ஜ் டைஜஸ்டர்ஸ் இருக்கு. அந்த ஏரியாவில் we can see the methanobacterium and also this methanobacterium are seen in the room and stomach of cattle. Cattle's stomachന് 4 chambers இருக்கு. அதில் ஒரு chamber தான் rumen. On the chamber, we can see this methanogens for cellulose digestion. Okay. And biogas is used for cooking and lighting. Cattle dung contain large amount of this methanobacterium. So the dung is used to produce biogas. This is called gobar gas. Okay. So cattle dung contain large amount of methanobacterium. So dung is used to produce biogas. This is called gobar gas. So this is a parts of a biogas plant. A concrete tank 10 to 15 feet deep. Concrete tank 10 to 15 feet deep to collect bio-wastes and slurry of the dung. There's a floating cover. There is a floating cover. placed over the slurry which keep on rising as the biogas is produced. Biogas produce ആവുന്ന ടൈമിൽ on the floating cover മേലേക്ക് പോകും. Otherwise what happen? The burst ആവാനുള്ള chance ഇരിക്കും. Okay. So here we are using a floating cover. And there is an inlet and there is two outlet. Through the inlet we provide the dung and water. Okay. Clear? Through the inlet we are providing dung and water. And there are two outlets. One is used to collect the biogas and other is used to collect the spent slurry. Spent slurry. Outlet to supply biogas, outlet to remove the spent slurry. And they are used as a biofertilizers. So this is a biogas plant. So a concrete tank 10 to 15 feet depth. And there is a floating cover. There is one inlet through which we are providing dung and water. And there are two outlets. One outlet is to supply biogas and another outlet to remove spent slurry. And this is used as a biofertilizer. Clear? This is a biogas plant. See, Indian Agriculture Research Institute, very, very important IARI. Okay, the Indian Agricultural Research Institute and Khadi and Village Industries Commissions developed the technology of biogas production in India. This can be framed as a question. What is IARI? Indian Agricultural Research Institute. What is KVIC? Khadi and Village Industries Commission. Okay, they developed the technology of biogas production in India. IARI and KVIC. What is IARI? Indian Agriculture Research Institute. What is KVIC? Khadi and Village Industries Commissions. Next, microbes as biocontrol agents. What do you mean by biocontrol agents? Biocontrol is a use of biological methods for controlling plant diseases and pests. നമുക്ക് தெரியும் ഒരു പെസ്റ്റിന് ഒരു നാച്ചുറൽ എനിമി ഇருக்கு. ഒരു നാച്ചുറൽ എനിമിയെ യൂസ് பண்ணி നമ്മളൊരു പെസ്റ്റിനെ കൺട്രോൾ பண்ணணும். We are controlling a pest using its natural enemy. We are controlling a pest by using its natural enemy. Okay, this is called biological pest control or biocontrol. For example, ladybird is used to control aphids. Dragonflies are used to control mosquitoes. It's a very, very important. Okay, so ladybird, this is a ladybird. Beautiful insight. The ladybird is used to control aphids and dragonflies are controlled mosquitoes. Okay, yes, dragonflies are controlled mosquitoes and ladybird are used to control aphids. And there is a bacteria called Bacillus thuringiensis. This bacteria are used to control butterfly caterpillar. Okay, you will study more about this Bacillus thuringiensis in the chapter Biotechnology and its applications. Clear? BT cotton, BT brinjal. And the area we will study more about this topic. Bacillus thuringiensis. So Bacillus thuringiensis, this bacteria produces some toxin. This toxin kills some insect, especially butterfly caterpillar. To control butterfly caterpillar, we are using Bacillus thuringiensis. The dried spores of BT are mixed with water and sprayed on to vulnerable plants such as brassicas and fruit trees. These are eaten by the caterpillar. In their gut, the toxin is released and the larvae get killed. Okay. Scientists have introduced the Bacillus thuringiensis toxin gene into plants, example BT cotton. This topic we will study in biotechnology and its application. Okay, so Bacillus thuringiensis is a bacteria. This bacteria is used to kill or control butterfly caterpillar. This bacteria produces some toxins. It is called BT toxin. It's an insecticidal toxin that will kill the larvae of some insect. Next, trichoderma. Trichoderma, it's a fungus. And they are seen in the root ecosystem, very, very important. Okay, they are seen in the root ecosystem. Trichoderma, it's a fungus. They are seen in the root ecosystem and they control several plant pathogens. Very, very important. Okay. So trichoderma is a fungus. They are generally seen in the root ecosystem. Okay. And these trichoderma control several plant pathogens. Plant pathogens. Trichoderma. It's a fungus where we can see root ecosystem control some plant pathogens. Next, baculovirus, especially the genus nucleopolyheterovirus. Very important. Baculovirus. Baculovirus. The genus nucleopolyheterovirus. Nucleopolyheterovirus. And this baculovirus attack insect and other arthropods. Insect and other arthropods. It is suitable for species-specific and it's a narrow spectrum insecticidal applications and desirable in IPM. What is IPM? Integrated Pest Management Program to conserve beneficial insect. It's a very important microbe that is used as a biocontrol agent, baculovirus. The genus nucleopolyheterovirus. And this baculovirus attack insect and other arthropods and it is suitable for species specific. It will not kill some useful microbes. നമുക്ക് beneficial കൊണ്ട microbes, அந்த microbes ഒന്നുമേ ഇത് attack பண்ணலாது. Okay, they will not attack the useful microbes. So they are species specific and they have a narrow spectrum of insecticidal applications. And desirable in IPM, integrated pest management. Okay, to conserve a beneficial insect. So these are the important microbes used as biocontrol agents. So Bacillus thuringiensis to control butterfly caterpillar. Trichoderma, several plant pathogens. Trichoderma found in root ecosystem. And baculovirus, the genus nucleopolyheterovirus. Okay, attack insect and other arthropods. And they're also used in IPM. What is IPM? Integrated Pest Management. And they are species specific. And they have a narrow spectrum of insecticidal applications. Clear? So next, microbes as biofertilizers. Okay, what do you mean by biofertilizers? See, biofertilizers are organisms that enrich nutrient quality of the soil. It's very important. So biofertilizers are organisms that enrich, that increase and that enrich the nutrient quality of the soil. These microbes are called the biofertilizers. Example is bacteria, fungi, cyanobacteria. So all these are example for biofertilizers. So you know the rhizobium. Rhizobium is a symbiotic bacteria in root nodules of leguminous plants. You can see the root nodules. The root nodules of leguminous plants contain a symbiotic bacteria. Its name is called rhizobium. Rhizobium fix atmospheric nitrogen. Okay, rhizobium fix atmospheric nitrogen. Rhizobium is a symbiotic bacteria present in the root nodules of leguminous plants. And there is a free-living bacteria in the soil. This is called the Azospirillum and Acetobacter. Azospirillum and Acetobacter. Azospirillum and Acetobacter, they are example for free-living bacteria. Rhizobium is not free-living. Rhizobium is a symbiotic bacteria present in the root nodules of leguminous plants. But Azospirillum and Acetobacter, they are free-living nitrogen-fixing bacteria present in soil. So all these are example for biofertilizers. Next one is mycorrhiza. It's very important. What is mycorrhiza? Mycorrhiza is a symbiotic association of fungi with the plants. Especially எங்க ரூட்டில் தான் இருக்குது. Plants ரூட்டில் தான் இந்த association வருது. Okay. So mycorrhiza is a symbiotic association of fungi with the plants. Symbiotic association of fungi with the plants. Especially which fungus? Genus Glomus. Okay, so there is no mycorrhiza, but this is with the mycorrhiza. So generally mycorrhizal association is found in where? Root. Clear? So symbiotic association of fungi with plants. Generally, this association is found in root of large plants, higher plants. So the fungus get food from the plant. We know that fungus there is no chlorophyll. So the fungus get food from the plant. Tirumbi, the fungal symbiont perform the following. It absorbs phosphorus from the soil and passes into the plants. Clear? And this fungal symbiont give resistance to root-borne pathogens and tolerance to salinity and drought. Okay. And this fungal symbiont also give a overall increase in plant growth and development. So what is mycorrhiza? It is a symbiotic association of fungus with the root of higher plants. Clear? So on the fungus in the plant food provide pannum. Tirumbi, the fungus absorb phosphorus from the soil and passes into the plant. Okay. And also this fungal symbiont give a resistance to root-borne pathogens and a tolerance to salinity and drought. Okay. And this fungal symbiont also give overall increase in plant growth and development. So these are the benefit the plant get from the fungal symbiont. Clear? Yes. So mycorrhiza. Next is cyanobacteria. They are called blue-green algae. Cyanobacteria is also called blue-green algae. See the cyanobacteria. These are the cyanobacteria, blue-green algae. They are the autotrophic microbes. They fix atmospheric nitrogen. Okay, so they are used as biofertilizers. In paddy field, cyanobacteria serve as biofertilizers. It is also add organic matter to the soil and increases fertility. Clear? So microbes are used as biofertilizers. What is biofertilizers? Organisms that enrich soil fertility are called biofertilizers. Okay. Examples are rhizobium. Rhizobium is a symbiotic bacteria present in the root nodules of leguminous plants, which can fix atmospheric nitrogen. Then some free-living bacteria like Azospirillum and Acetobacter. They can also fix atmospheric nitrogen. Clear? And there is mycorrhiza. What is mycorrhiza? Symbiotic association of fungus with the root of higher plants. Okay. Fungus get food from the plant. Tirumbi and the fungus they absorb phosphorus and pass it to plants, and they provide resistance against salinity and drought, and also increase overall development of plants. Clear? And cyanobacteria, they are also called blue-green algae. They can also fix atmospheric nitrogen. Okay. In paddy field, this cyanobacteria serves as biofertilizers. Okay. So this is all about the chapter microbes in human welfare. Okay, first microbes in household products. And we mostly study lactobacillus and lab, they convert milk into curd. Okay, then Propionibacterium shermani, Swiss cheese. Penicillium roqueforti, Roquefort cheese. Okay, Saccharomyces cerevisiae, fermenting duff. Clear? Next, industrial purpose, Saccharomyces cerevisiae, fermented beverages. Okay, then some antibiotics. The first antibiotic is penicillin, discovered by Alexander Fleming. Okay, next is some organic acids. Aspergillus niger produces citric acid. Acetobacter aceti produces acetic acid. Clostridium botulicum produces butyric acid. And Lactobacillus produces lactic acid. Okay, and some enzymes, lipases, used in detergent formulation to remove oily stain from the laundry. Pectinase and proteases, they are used to clarify bottled juices. Streptokinase, clotbuster, taken from the bacteria Streptococcus. Okay. Then some bioactive molecules, cyclosporine, immunosuppressive agent, taken from the fungus Trichoderma polyspora. Then statin, cholesterol lowering agent. We know that sewage treatment, primary treatment and secondary treatment. Primary treatment will 2 steps, sequential filtration and sedimentation. Okay, clear? What is flux? Bacteria associated with the fungal filaments are called flocks. If BOD அதிகமாயால் water pollution அதிகமாக இருக்கும், BOD கம்மியாயால் water pollution கம்மியாக இருக்கும். BOD is a measure of amount of organic content in the water. Clear? So this is all about this chapter. It's an important chapter for NEET exam. Study well. Okay. Thank you. See you again. Bye.